Assalamualaikum and salam sejahtera to our lecturer Dr. Muhammad Nizam bin Zahari. We are from Group C. We present about cell culture.
First of all, for introduction, what is cell culture? Cell culture refers to the removal of cells from an animal or plant and their subsequent growth in an artificial environment. Next, basic stage in cell culture. Number 1 is primary cell culture. Primary culture refers to the stage of the culture after the cells are isolated from the tissue and proliferated under the appropriate conditions. Number 2 is cell line. After this first subculture, the primary culture becomes known as a cell line or subclone. And number 3 is cell strain. If a subpopulation of a cell line is positively selected from the culture by cloning or some other method, this cell line becomes a cell strain. As for culture conditions, the artificial environment where the cells are cultivated varies greatly depending on the cell type, but it commonly requires a suitable vessel containing a substrate or medium that supplies the nutrients, for example, amino acids, carbohydrates, vitamins, and minerals. Secondly, physical-chemical environment, for example, pH, osmotic pressure, and temperature. And the last one is growth factors, hormones, and gases such as oxygen and carbon dioxide. And the last part in introduction is application of cell culture. Cell culture can act as excellent model system for the normal physiology, cell biology, and biochemistry of cell, and the effect of drug, radiation, and toxic compound on the cells, and the study of mutagenesis and carcinogenesis. Next, cell culture also can be used for gene transfer studies.
Safety in the cell culture laboratory consists of several things such as biosafety levels, which consists of PSL1 for basic level of protection common to most research and clinical laboratories in humans, PSL2 for moderate risk agent known to cause human disease of varying severity by injection or mucous membrane exposure, PSL3 appropriate for indigenous or exotic agent with a known potential for aerosol transmission, and lastly PSL4 for exotic agents that pose a high individual risk of life-threatening disease by infectious aerosols and for which no treatment is available. Next, safety equipment consists of biosafety cabinets, enclosed containers, and PPE. Example of PPE such as glove, laboratory coats, boots, respirators, face masks, and shield. Lastly, safe lab practice. One should have always wear appropriate personal protective equipment such as change gloves when contaminated, wash hands after working with hazardous material and before leaving the laboratory. Next, decontaminate all work surfaces before and after experiment, and lastly, decontaminate all potentially infectious materials before disposal. Next, we move to cell culture equipment. The specific requirement of a cell culture lab depend mainly on the type of research conducted. For example, the need of mammalian cell culture laboratory is different from insect cell culture laboratory. However, all cell culture laboratories have the common requirement of being free from pathogenic microorganisms. The equipment that must have in the cell culture laboratory, such as basic equipment, as example, cell culture hood, incubator, water bath, centrifuge, refrigerator and freezer, and sterilizer. Other than that, expanded equipment such as aspiration pump, pH meter, confocal microscope, and flow cytometer. There's also additional supplies such as cell culture vessels, pipettes and pipetters, syringe and needles, waste container, media, sera and reagent, and lastly, cells. Cell culture laboratory must have the septic work area, which is for sterile handling, incubation, and storage of cell cultures, reagent, and media. Next, cell culture hood. The Class 1 cell culture hood are significant levels of protection to laboratory personnel and to the environment, but they do not provide cultures protection from contamination. Class 2 cell culture hood designed for work involving BSL 1, 2, and 3 materials and also provide an aseptic environment necessary for cell culture experiment. Class 3 cell culture hood are gas tight and they provide the highest attainable level of protection to personnel and the environment. A Class 3 biosafety cabinet is required for work involving non-human pathogens and other BSL4 materials. And lastly, airflow cell culture hood, which is to protect the working environment from dust and other airborne contaminants by maintaining a constant unidirectional flow of HEPA filter air over the work area. Next, the cell counter hood layout. It should be large enough to be used by one person at a time, be easily cleanable inside and outside, have adequate lighting, and be comfortable to use without requiring a good position. Here are the example of cell counter hood layout. The arrangement of items within the cell culture hood must a wide, clear workspace in the center with cell culture vessel. Next, pipette in the front right where it can be reached easily. The reagents and media must be in the rear right to allow easy pipetting. Tube rack in the rear middle holding in additional reagents. And lastly, the small container must be in the rear left to hold liquid waste.
In addition, in cell culture laboratory also have storage that's specific for each equipment such as for chemicals, liquid, consumable, glassware, specialized equipment, tissue, and cell. Each of these equipment must be stored properly according to the instruction on the label. In storage also have refrigerator where it consists of domestic refrigerator which is for storage equipment such as media and reagent at 2 to 8 degrees Celsius and large refrigerator which is restricted for cell culture. For refrigerator, it's important to make sure to clean the refrigerator regularly to avoid contamination. Moreover, a cryogenic storage where it functions to store cell line in continuous culture, for example, working stock of the cell that require it to preserve them. There consists of two types which are vapor-based where it's to minimize the risk of explosion with cryo storage tube and required for storing biohazardous material, while the another one is liquid-based, usually have longer static holding time and are therefore more economical. Lastly, a cell counter where essential for quantitative growth kinetic and useful more than 2 or 3 cell lines are cultured in the laboratory. In cell counter, automatic cell counter is used to measure cell count and viability and accurately and precisely in less than a minute per sample. Next is aseptic technique. Aseptic technique is important to maintain a clean and safe culture environment in order to prevent contaminating microorganisms from the environment from entering the culture. Or in other words, it is to provide a barrier between the microorganism in the environment and the sterile cell culture. There consists 4 elements in aseptic technique. Firstly is dry work area. For example, cell culture hood, which should be properly set up and be located in area that is restricted to cell culture, which aim to reduce contamination from airborne pesticide and aerosol. To make sure work area is dry, before and after use, the work surface should be disinfected thoroughly and the surrounding area and equipment should be cleaned routinely by using 70% of ethanol. Secondly is good personal hygiene, where it required to wash hands before and after working with cell culture and wearing personal protective equipment, which is PPE, to protect him from hazardous material and also to reduce the probability of contamination from shed skin as well as dirt and dust from clothes. Thirdly is dry handling. For example, by avoid pouring media and reagent directly from bottle or flask, or avoid talking, sneezing, or coughing, and use different pipette for different reagent and media to avoid cross-contamination. And lastly is dry reagent and media. By always try any reagent, media, or solution, where in here it means that any equipment that prepared in the laboratory must be dried by using the appropriate sterilization procedure, for example, autoclave and dry filter. Here is the example of accepted technique checklist. Biological contamination. Contamination of cell culture is most common problem encountered in cell culture laboratories, sometimes with very serious consequence. Here is the major biological contamination. First is bacteria. Due to their ubiquity, size, and fast growth rate, it is easily to contaminate in cell culture. Bacterial contamination is easily detected by visual inspection of the culture, where infected culture usually appear cloudy or turbid owing to the change in pH caused by the infection. Under a low-power microscope, the bacteria appears a tiny, moving granule between the cells. Second is yeast. Like bacterial contamination, culture contaminated with yeast become turbid, especially if the contamination is in an advanced stage. Change in pH also cause contamination become heavy. Under microscopy, yeast appear as individual ovoid or sprinkle particles that make part of smaller particles. Third is mold. The culture become turbid due to the increased pH after contamination appear. Under microscopy, the MUZILAH usually appear as thin, yeast-like filament and sometimes as denser clump of spore. Then is virus. It is a serious health hazard. Viral infection of cell culture can be detected by electron microscopy, immunostaining with a panel of antibody, ELISA assay, or PCR with appropriate viral primer. Lastly is mycoplasma. Decreased rate of cell proliferation, reduced saturation density, and agglutination in suspension culture. Thus, it is difficult to detect until they achieve extremely high density and cause the cell culture to deteriorate. This contamination can detect in using fluorescence staining, ELISA, PCR, immunostaining, autoradiography, or microbiological assay.
Next slide is covered on cell culture basics. Cell culture basics provides information on the fundamentals of cell culture, including the selection of the appropriate cell line for experiments, media requirements for cell culture, adherent versus suspension culture, and morphologies of continuous cell lines. For cell lines, there will be two components under it, which is selecting appropriate cell line and how to acquire cell line. In selecting appropriate cell line, few things must be considered, which is species, functional characteristic, finite of continuous, normal or transformer, growth conditions and characteristics, and other characteristics. Let me explain two examples from selecting the appropriate cell line, which are species and functional characteristics. For species, usually non-human and non-primate cell lines have fewer biosafety restrictions, but ultimately the types of experiments will dictate whether to use species-specific cultures or not. For functional characteristics, it covers things such as what is the purpose for the experiments. For example, liver and kidney-derived cell lines may be more suitable for toxicity testing. Next is culture environment. Culture environment is considered as major advantages of cell culture due to the ability to manipulate psychochemical and physiological environment. Under culture environment, there are few components can be included such as media, pH, CO2 and temperature. For media, it provides the necessary nutrients, growth factors and hormones for cell growth, as well as regulating the pH and the osmotic pressure of the culture. There are three basic classes of media, which include basal media, reduced serum media, and serum-free media. They are different in requirements for supplementation with serum. For basal media, the majority of cell lines grow well in basal media, which contain amino acids, vitamins, inorganic salts, and a carbon source such as glucose. But these basal media formulations must be further supplemented with serum. For usage of reduced serum media, it is considered as another strategy to reduce the undesired effects of serum in cell culture experiments. Reduced serum media are basal media formulations enriched with nutrients and immune-derived factors, which reduce the amount of serum that is needed. Lastly is serum-free media. Serum-free media circumvents issues with using animal sera by replacing the serum with appropriate nutritional and hormonal formulations. Serum-free media formulations exist for many primary cultures and cell lines, including recombinant protein-producing lines of Chinese hamster ovary, where the subdermal cell lines, the insert lines SF9 and SF21, and for cell lines that act as hosts for viral production and others. One of the major advantages of using serum-free media is the ability to make the medium selective for specific cell types by choosing the appropriate combination of growth factors. Next component is pH. Most normal mammalian cell lines grow well at pH 7.4, and there is very little variability among different cell strains. Some transformed cell lines have been shown to grow better at slightly more acidic environments, such as pH 7.0 to 7.4. Some normal fibroblast cell lines prefer slightly more basic environments, such as pH 7.4 to 7.7. Next component is carbon dioxide. The growth medium controls the pH of the culture and buffers the cells in culture against changes in the pH.
Next, in this slide, I will talk about one of the components under cell culture environment, which is temperature. Optimal temperature for cell culture largely depends on the body temperature of the host from which the cell was isolated and to a lesser degree on the anatomical variation in temperature. For human and mammalian cell line, they are maintained at 36 to 37 degrees Celsius for optimal growth. For insect cell culture, the optimal growth is 27 degrees Celsius, while for avian cell line, they require 38.5 degrees Celsius for maximum growth. And lastly, for cold-blooded animal, they range between 15 degrees Celsius to 26 degrees Celsius. Next, in this slide, I will discuss about cell morphology, examining the morphology of the cell in culture. For example, their shape and appearance considered as essential for successful cell culture experiment. In addition to confirming the healthy status of cell, inspecting the cell by eye and microscope each time they are handled will allow for detection of any sign of contamination early on and to contain it before it spread to other culture around the lab. Signs of deterioration of cell include granularity around the nucleus, detachment of the cell from the substrate, and cytoplasmic inoculation. Signs of deterioration may be caused by a variety of reasons, including contamination of the culture, senescence of the cell line, or the presence of toxic substance in the medium, or they may simply imply that the culture needs a medium change. Allowing the deterioration to progress too far will make it irreversible. There are three variations in mammalian cell morphology, which include fibroblastic, epithelial-like, and lymphoblastic. For fibroblastic or non-fibroblastic, the cells are bipolar or multipolar and have elongated shape. They grow attached to a substrate. Next, for epithelial-like, the cells are polygonal in shape with more regular dimension and grow attached to a substrate in discrete patch. Lastly, for lymphoblast-like cells, they are spherical in shape and they are usually grown in suspension without attaching to a surface.
Traits. There are guidelines for maintaining cultured cells, which provide guidelines and general procedures for routine subculturing, thawing and freezing of cells in culture. Subculturing, also referred to as passaging, is the removal of the medium and transfers of cells from a previous culture into fresh growth medium. Cell growth in culture proceeds from the lag phase following seeding to the log phase, where the cells proliferate exponentially until exceed the capacity of the medium. So when to subculture, there are three criteria considered before subculturing, which is cell density, medium exhaustion, and subculture schedule. First one, cell density. Adherent culture should be passage when they are in the log phase before they reach confluence. Normal cells stop growing when they reach confluence and it takes them longer to recover when reseed. Transformed cell can continue proliferating even after they reach confluence, but they usually deteriorate after 2 doublings. Similarly, cells in suspension should be passage when they are in log phase growth before they reach confluency. When they reach confluency, cell in suspension clump together and the medium appears to beat when the culture flask is swirled. Next, a drop in pH of the growth medium usually indicates a build-up of lactic acid, which is a byproduct of cellular metabolism. Lactic acid can be toxic to cells and decreased pH can be suboptimal for cell growth. You should subculture your cell if you observe a rapid drop in pH with an increase in the cell concentration. Next, for subculture schedule, passaging your cells according to a strict schedule ensure reproducible behavior and allows you to monitor their health status. Vary the seeding density of your cultures until you achieve consistent growth rates and yield appropriate for your cell type from a given seeding density. Unhealthy culture and improper functioning of culture system can deviate growth pattern.
Culturing adherent cell. Material needed for this method: culture vessel containing adherent cell, 37 incubator with humidified atmosphere of 5% carbon dioxide, complete growth medium, pre-warm to 37 degrees Celsius, and so on. For protocol, remove the spent cell culture media from the culture vessel, then wash using a balanced salt solution. Add water solution to the opposite of at the cell layer and rock the vessel back and forth. Then remove the water solution from the culture vessel, add pre-warm dissociation reagent to side of flask, rock the container, incubate at room temperature about 2 minutes, observe the cell under the microscope for detachment. After 90% of the cell have detached, take the vessel to a low drain, add the equivalent of 2 volume of pre-warm complete growth medium, disperse the manure by pipetting over cell surface, transfer cell to a 50 ml conical tube and centrifuge at 200 speed for 5 to 10 minutes. Suspend the cell pellet in minimal volume of pre-warm complete growth medium and remove cell pearl for counting. Dilute cell suspension to the recommended seeding density for the cell line and pipette the volume into new cell culture vessel and return the cell to the incubator.
The next method is subculturing suspension cells. Passaging suspension cultures is one of the steps in subculturing suspension cells. There are two ways to passage the suspension, which are by diluting the cells in culture flask and continue expanding them, or removing a portion of the cells from the culture flask and diluting the remaining cells to the appropriate seeding density for the cell line. Suspension culture vessel is a step that allows for better gas exchange and the culture of larger volume of cells. There are two types of flasks, which are shaker flask and spinner flask. And for the spinner flask, there are two models, which are spinner flask with hanging stir bar and spinner flask with vertical impeller. Materials needed include, firstly, culture vessels with suspension cells. Second, shaker flask without buffers or spinner bottles. Third, complete growth medium pre-warm to 37 degrees Celsius. Fourth, 37 degrees Celsius incubator with humidified atmosphere of 5% carbon dioxide. Fifth is magnetic stir plate, roller rack or shaking platform. And the sixth is reagents and equipment to determine viable and total cell counts. The next one is protocol for passaging suspension cells. For this protocol, all solutions and equipment that come in contact with the cells must be sterile. Next, when the cells are ready, the flask is removed from the incubator and a small sample is taken out from it. Then the total number of cells and present availability is determined from the sample. The needed volume of media is calculated to be added into the culture. Appropriate volume of pre-warm growth medium is added into the culture flask. And lastly, the caps is loosened one full turn to allow for proper gas exchange and return the flask to the shaking incubator. There are some recommendations that need to be concerned when culturing the cell. Firstly, in shaker flask, make sure that the shaker flask doesn't have buffers, and also centrifuge the cell suspension at 100 times gram for 5 to 10 minutes and suspend the cell pellet in fresh growth medium once every 3 weeks. And for the spin flask, ensure that impeller mechanism rotate freely. The top of the pedals should be slightly above the medium, and also centrifuge the cell suspension at 100 time gram for 5 to 10 minutes, and resuspend the cell pellet in fresh growth medium once every 3 weeks.
Next, cell culture method, which is freezing cell. Established cell line is valuable resource and replacement is expensive and time-consuming. So it's very important they are frozen down and preserved for long-term storage. It's available from subculturing, it should be frozen, stored, protected, and not be made available for general laboratory use. So the best method is storing them in liquid nitrogen in complete medium in the presence of cryoprotective agents such as DMSO. Okay. Guideline for freezing cell. All solution and equipment must be sterile, stored below -70 degrees Celsius because it's beginning to deteriorate above 50 degrees Celsius. Freeze at a high concentration at a lower passage number as possible. Lastly, freeze the cell slowly by reducing the temperature at approximately 1 degree Celsius per minute using a control rate cryofreezer. For protocol, prepare freezing medium and store at 2 degree and to 8 degrees Celsius until used. For hydrogen cell, gently detach cell from tissue culture vessel, suspend the cell in the complete medium required for that cell type. Next, determine the total number of cells and preserve viability using hemocytometer and so on. Centrifuge the cell suspension at approximately 100-200 for 5 to 10 minutes without disturbing the cell pellet. Then resuspend the cell pellet in cool freezing medium as recommended. Dispense aliquots of the cell suspension into cryogenic storage vial. Aliquot them frequently and gently mix the cell to maintain a homogeneous cell suspension. Next, freeze the cell in control rate freezing apparatus, decreasing the temperature approximately 1 degree Celsius per minute. Last but not least, transfer frozen cell to liquid nitrogen and store them in the gas phase above liquid nitrogen.
For towing frozen cells, materials needed are as stated. Protocol for towing frozen cells. First, remove the cryovial containing frozen cells from liquid nitrogen storage and immediately place it into water bath. Quickly thaw the cells by gently swirling the vial in the water bath until there is just a small bit of ice left in the vial. Transfer the vial into a laminar flow hood. Before opening, wipe the outside of the vial with ethanol. Next, transfer the thawed cell dropwise into the centrifuge tube containing the desired amount of pre-warm complete growth medium appropriate for your cell line. Centrifuge the suspension at approximately 200 times for 5 to 10 minutes. The actual centrifugation speed and duration varies depending on the cell type. After centrifugation, check the clarity of supernatant and visibility of a complete pellet. Aseptically decant the supernatant without disturbing the cell pellet. Gently resuspend the cell in complete growth medium and transfer them in the appropriate culture vessel and into the recommended culture environment. Recommendations for towing frozen cells. Thaw frozen cells rapidly in 37-degree water bath. Dilute the thawed cells slowly using pre-warm growth medium. Plate thawed cells at high density to optimize recovery. Always use proper aseptic technique and work in a laminar flow hood. Always wear personal protective equipment. Handle regions containing DMSO using equipment and appropriate practice.
The last method is support protocols. Counting cells in hemocytometer is one of the support protocol. To count the cells, firstly, by using alcohol, clean the chamber and the cover slip, and then fix the cover slip in place after it has dried. Second, harvest the cells by adding 10 microliter of the cells to the hemocytometer. Next, place the chamber in the inverted microscope under time magnification objective. And lastly, count the cells in the centrifugal square and multiply by 10 power 4 to estimate number of cells per milliliter. For trypan blue exclusion, it aims to determine the cell viability. And to use this solution in support protocol, firstly, determine cell density by using hemocytometer. Then, prepare 0.4% of trypan blue in isotonic salt solution. Next, add 0.1 ml of trypan blue stock solution to 1 ml cells. The fourth step is load hemocytometer and examine under microscope at low magnification. And lastly, count the number of blue staining cells and number of total cells by using the formula below. Finally, is concentrating cells. It aims to concentrate cells from a suspension culture. To concentrate cells, firstly, transfer the cell suspension to a centrifuge tube and centrifuge for 10 minutes. Second, remove the supernatant. Then add fresh medium to the side of the tube and pipette up and down to resuspend the cell pellet. Lastly, transfer the cells to a container.
Here the list of our reference.